which of the following are the reasons for occurrence of multi-drug resistance in microbial pathogens in India? Right? So, what are the reasons for multi-drug resistance in microbial pathogens in India? That is the question. That means this question is related to antimicrobial resistance. Right? So, this question is related to antimicrobial resistance. So, first of all, we should understand what do we mean by antimicrobial resistance. Antimicrobial resistance is resistance shown by certain pathogens towards certain drugs. Those drugs earlier were used to cure or were used to treat the diseases caused by those pathogens. But with antimicrobial resistance, those drugs are now ineffective against those pathogens. And as a result of this, we will have to use certain other drugs for the treatment of those pathogens now, because the drugs which were used earlier have become ineffective now. And there are many reasons for this antimicrobial resistance, which is considered as one of the threats in world as well as in India. And the antimicrobial resistance is defined by WHO as AMR occurs when bacteria, viruses, fungi and parasites change over time and no longer respond to medicines, making infections harder to treat and increasing the risk of disease spread, severe illness and even death. Right? So that is how antimicrobial resistance is defined by WHO, World Health Organization. Moreover, as a result of drug resistance, antibiotics and other antimicrobial medicines become ineffective and infections become increasingly difficult or impossible to treat. Why they become difficult? Because the drugs which we were using earlier for their treatment are no longer effective. And now, if the drugs which are used for those treatment are not available at all, then it will become impossible for us to treat those infections. Right? So that's why AMR is considered as a health threat by WHO. The question is about the reasons for antimicrobial resistance in various pathogens, especially in India. So, first statement is genetic predisposition of some people. So, this is not the reason because of which antimicrobial resistance or multi-drug resistance will occur in pathogens, because genetic makeup of or genetic predisposition of certain individuals is not at all related with the antimicrobial resistance, and that's why first statement is incorrect statement. Second, taking incorrect doses of antibiotics to cure diseases, right? So this is one of the important reasons for antimicrobial resistance, because what happens often during treatment of certain disease, we use antibiotics, and antibiotics are supposed to take for certain duration. It may be for 3 days, 5 days or 7 days. But suppose if we take antibiotics for first day and we started feeling better, there may be a case that we no longer continue the dose of antibiotics for the prescribed time. And that's why the strains of pathogens which are still there in our body develop resistance against the drugs that we have used or the antibiotics that we have used on the first day, right? So the improper or incorrect doses of antibiotics is one of the prime reasons for antimicrobial resistance or multi-drug resistance, right? So underuse as well as overuse of antibiotics lead to multi-drug resistance, and that's why second statement is correct statement. Third, using antibiotics in livestock farming. So this is also a correct statement, because in livestock farming, in order to increase the production, often the farmers use medicines, and these medicines are nothing but antibiotics. And as a result of the use of antibiotics in livestock farming, the strains of those antibiotics enter in our bodies as well. As a result of which, the bacteria, the pathogens, develop resistance against the overused antibiotics in the livestock farming, right? So this is also the reason for AMR or multi-drug resistance. Fourth, multiple chronic diseases in some people. So this is also a correct statement, because if there are chronic diseases in, or multiple chronic diseases in few people, then the use of medicines, the use of drugs by these people will be more. And as a result of this, there will be increasing tendency in the pathogens in the body of those people to develop resistance against those medicines that have been used in excess, right? So that's why even fourth statement is correct statement, and that's why second, third and fourth statement are correct, and hence answer of this question is 2, 3 and 4, option D, right? So apart from these reasons, there are some other reasons for antimicrobial resistance as well. So we will discuss those reasons of AMR. So the main drivers of antimicrobial resistance include: first, misuse and overuse of antimicrobials, which includes antivirals, antiparasites, antibiotics as well, right? So first is the misuse. Second, lack of access to clean water, sanitation and hygiene for both humans and animals. So if there is lack of clean drinking water, sanitation, etc., then there will be a chance that these people will fall ill, and that's why they will use medicines, and the medicines may not be used in an effective manner or proper manner. And that's why even second is one of the important reasons. Second statement is one of the important reason for antimicrobial resistance. Next, third, poor infection and disease prevention and control in healthcare facilities and farms. So that is also a reason, because in hospital settings there will be patients, and there is a tendency in the pathogens to jump from ill patient to healthy patient or healthy person in the vicinity of that hospital, and that's why there will be a spread of antimicrobial resistance through poor infection and disease prevention in the hospital settings. Next, poor access to quality, affordable medicines, vaccines and diagnostic. So if there is lack of availability of healthcare, then there will be over the counter sale of medicines, and that will lead to improper use of antibiotics, and that's why this is one of the important reasons for AMR. Next, lack of awareness and knowledge, as a result of which there will be underuse or overuse of medicines. Next, lack of enforcement of legislation. So there are legislation which make sure that there is no over the counter sale of various antibiotics, but those laws are not enforced properly, and as a result of which the medicines are made available even without doctor's prescription, and that leads to improper use of antibiotics, right? So all these are the important reasons, and the answer for this question that we have discussed is D. Next question is, RNA interference, that means RNAi. RNA interference technology has gained popularity in the last few years. Why? Right? So, what is RNA interference? So, RNA interference or RNAi is a technology based upon which we can make sure that the manifestation of certain characteristics related to certain genes can be curbed, right? That means we can make sure that the proteins synthesized based upon the information on genes are prohibited from being synthesized. As a result of this prohibition of protein synthesis, what will happen? The characteristics which are related to those proteins will not be seen in that particular individual. And this is one of the important methods that is naturally used by various organisms, be it plants, fungi, even animals, to make sure that they develop resistance to certain pests. There is lack of effectiveness of attack by certain pests on these plant species, especially because of RNA interference technology, right? So let me read the definition for RNA interference. So what is RNA interference? RNAi, a regulatory system occurring within the eukaryotic cells, that means those cells which have clearly defined nucleus. So it is a kind of regulatory system that occurs in these eukaryotic cells that control the activity of genes. So what RNAi do? It controls the activity of genes. And as a result of this control over the activities of genes, we can make sure that the protein that is synthesized by those genes is prohibited from getting synthesized. Next, RNAi functions specifically to silence or deactivate genes, right? So with RNAi, what we can do, we can either silence or deactivate certain genes and thus the proteins synthesized by those genes. In a wide variety of organisms, including animals, plants, fungi, RNAi is triggered by double-stranded RNA, that means dsRNA. Next, and where all this RNAi technique or technology is used, RNAi technology is used for disease control. That means if certain genes are vulnerable to a specific disease, then we can make sure that those genes are silenced so that the vulnerability to that disease can be controlled, right? So for disease control. Second, production of animals of commercial interest. So if there is a particular gene that is leading to lack of production of, let's say, milk after some time in a particular buffalo, then we can silence that gene. We can make sure that that gene is deactivated so that milk production is not curbed or not reduced in that buffalo, right? So that is also one of the use of RNAi technology. Next, production of animal models for research use. So that is also one of the use of RNAi, because based upon RNAi, we can make sure that the animal models are resembling to human beings, and that can be achieved if we can silence or we can deactivate certain genes in the animal's body, right? So that's why even third is one of the important application. Next, RNAi has provided a way to control pests and diseases, introduce novel plant traits, and increase crop yield as well. So one of the most important application of RNA interference is to make plants resistant to certain kind of pest and those diseases, right? So these are the applications. Now let us discuss what are the statements that are being given in the question. based upon which we can easily find answer now. So the first statement is, it is used in developing gene silencing therapies, right? So the very idea behind RNA interference is gene silencing or genes deactivating, and hence first statement is correct statement. Second, it can be used in developing therapies for treatment of cancer, right? So this is also the application, because based upon RNAi, certain genes, if we found that certain genes are vulnerable to the cancer, then those genes can be silenced, and hence it can be used for the treatment of cancer as well. Third, it can be used to develop hormone replacement therapies, right? So with RNA interference, it would be difficult, or it is not possible for us to replace hormones inside the bodies of organisms, and that's why this statement is incorrect with respect to RNA interference, because RNA interference is not about replacement of hormones. If we want to silence the generation of certain hormone in the body of an organism, then that is possible with RNA interference, but replacement is not possible. Next, it can be used to produce crop plants that are resistant to virtual pathogens. So this is correct statement. So answer for this should include first, second and fourth. So first, second and fourth is mentioned in option A, and hence answer of this question is A, right? So since last few years, there is increasing interest by UPSC that we are seeing in the biotechnology, right? So biotechnology should be the focus of your studies. You should be very thorough with biotechnology, and this is quite interesting chapter which we will deal in our regular sessions, right? So let us discuss next question now. What is Cas9 protein that is often mentioned in the news, right? So you must be aware of one of the important gene editing tool, CRISPR-Cas9. CRISPR-Cas9. So Cas9, they have asked, what is Cas9 protein? What does it do? So Cas9 protein is used to carry out genetic editing in the targeted site, right? So that's why Cas9 can be used as a molecular Caesar in the targeted gene editing, and that's why answer of this question is A, right? So Cas9 is a protein which is used to carry out gene editing at the specific site. So Cas9 protein is an RNA guided endonuclease that can be used for the site specific cleavage of double stranded DNA, right? So based upon the Cas9 protein, we can target a particular site called as cleavage site on DNA, and we can carry out the cut at that particular site, right? So that is the use of Cas9 protein. Next now. Next question is, with reference to recent development in science, which one of the following statement is not correct? So out of these statements, which one is not correct? First, or A, functional chromosomes can be created by joining segments of DNA taken from cells of different species, right? So this is not correct, because till now we have not developed a functional chromosome based upon the genes derived from cells of different species, or DNAs taken from cells of different species. This experimentation we have not achieved as of now, and that's why this is not correct. Next, B, Pieces of artificial functional DNA can be created in laboratories. So we are able to create the pieces of artificial functional DNA because of the knowledge that we have got about the DNAs through programs like, or through projects like human genome projects, right? So second statement is correct statement. C, a piece of DNA taken out from an animal cell can be made to replicate outside a living cell in the laboratory. So this is also a correct statement, because we can replicate, or it is possible for us to replicate the piece of DNA inside the laboratories. Last option, cells taken out from plants and animals can be made to undergo cell division in the laboratory petri dishes. So this is the age old phenomenon that we are aware of, and that's why out of these four options, option A is not correct, and that's why that is our answer. So answer for this question is